Okay, good morning, class, and welcome to this first official lecture in journal microbiology. Here we are going to look at the methods used by microbiologists to classify microorganisms. I say microorganisms, but I will talk a little bit about general classification. That will include all organisms, especially now the microbes. So microbial classification is a little more complex than the general taxonomy you're familiar with in zoology and botany, because here we're dealing with organisms that need more detailed studies. And the typical method of taxonomy ranking that we have might be difficult or problematic employing here. So I would like to give a little bit of history on how we got to where we are today. We are familiar of the typical two-kingdom classification that was proposed, since 1773, wherein all organisms were either classified as plants or as animals. But more studies revealed that there were some organisms that were neither plants nor animals, which means that something had to be done to accommodate them. So in 1986, a German zoologist suggested that a third kingdom be considered, which he named Protista. And to him, these Protista should be organisms that were unicellular and that could neither be classified as plants nor animals. So to him, it should include what he called lower protists and higher protists. And lower protists to him were more simple, smaller organisms like bacteria, while higher protists were a little more larger organisms, and included algae, fungi, and protozoa. But the biggest problem we had, or that his system experienced, was that there was no basis. He had put forward no grounds for separating organisms as lower protist or higher protist. So the reason why he said bacteria should be lower protist and protozoa should be higher protist was not forthcoming. And therefore, it was just for convenience. But thank God to evolving science with the development of the powerful magnification offered by the electron microscopy in the late 1940s, more detailed studies could be done on the structure and organization of organisms. And therefore, scientists were able to identify that organisms previously classified as lower protists had organelles without membranes. So typically when they observe a bacterial cell, they realize that a bacterial cell was not enclosed in any membrane, while other organisms that were placed under the higher protists, remember we're talking of fungi, protozoa, and algae, typically had membrane-bound organelles. So this was now a reason for classifying organisms as lower protists and as higher protists. Okay, so scientists now went further to name these lower protists, designate them as prokaryotic, and higher protists were designated as eukaryotes. So we are showing that bacteria were classified, therefore, as what? Prokaryotic organisms, while fungi, algae, and protozoa were classified as eukaryotic organisms. A more comprehensive system that brought forth the typical 5-kingdom classification system that you are very familiar with was proposed by Whitaker in 1969. And this system was more revealing in terms of the basis for the classification. To Whitaker, organisms had evolved into 3 similar levels of cellular organization that accommodated 3 principal modes of nutrition. So we have organisms that are evolved to be photosynthetic in their mode of nutrition. We had organisms that had evolved to be ingestive in their mode of nutrition, and we had organisms that were absorptive in their mode of nutrition. So based on this, it was convenient to place organisms under the 5-kingdom classification. We had first the prokaryotic organisms that had principally the photosynthetic mode of nutrition. Some had absorptive mode of nutrition. So we had now the prokaryotes named under the kingdom Monaria. But typical of this is that they lack the ingestive mode of nutrition. That is very important because ingestion comes with the mouth. Unicellular organisms that had almost all exhibited all 3 modes of nutrition, either absorptive, ingestive, or photosynthetic, were placed under the kingdom. And this included the fungi, sorry, this included the algae and the protozoa. This included the algae and protozoa. And remember here, we're showing that they have the ability to undertake all 3 principal modes of nutrition. While multicellular and multinucleate organisms, remember we are under eukaryotes, that were either photosynthetic were placed as higher plants, were placed under the kingdom Plantae and included higher plants, green lower plants, and every other plant that can undergo photosynthesis. Those that had ingestive mode of nutrition, we're talking of multicellular, multinucleate organisms that had ingestive modes of nutrition were classified as animals, while higher plants that had the absorptive mode of nutrition were placed under the kingdom fungi. So we're saying that the 5-kingdom classification had 1, the kingdom Monaria, which had what? Bacteria, which had photosynthetic and sometimes absorptive mode of nutrition. We had the kingdom Protista. This Protista had what? Both absorptive, ingestive, and photosynthetic mode of nutrition, and included microalgae and protozoa. We also had organisms that were multinucleated, had absorptive mode of nutrition, and were multicellular, and were placed under the kingdom fungi and included the yeast and the molds. And finally, microorganisms that were multinucleated, multicellular, had principally the photosynthetic mode of nutrition. These were placed under the kingdom Plantae, and microorganisms that were multicellular, multinucleated, and had principally the ingestive mode of nutrition. These were placed under the kingdom Animalia. So here we have 5 kingdoms as such. But again, studies bring more problems. With more advancement in method of studies, it was discovered that the kingdom Monaria had far more diverse organisms to be accommodated in one kingdom. The organisms that were too diverse, and Berger's Manual of Evolutionary Biology came out with the notion of domain. So the kingdom Monaria had organisms that were lacking membrane-bounded organelles. That was okay and fine. But what was initially common among these organisms was that all of them were bounded with a peptidoglycan layer. All of them were bounded with a peptidoglycan layer. But with more studies, it was revealed that that was not the case. There were organisms that lacked the peptidoglycan layer. And these organisms had varied mode of nutrition. Some were photosynthetic using light, some were able to manufacture their own food using chemicals and with different variation in cellular organelles and level of organization. So these organisms were placed under the domain Archaea and identified under the kingdom Archaeobacteria. Because they had organisms that were not very unique, they lack a peptidoglycan layer, they were also identified in very, very extreme environments. These organisms could survive under the hot springs, acid lakes, and salty waters. So it was difficult to place them under the normal organisms that could be pathogenic to you and I. So they were identified under a domain on its own, domain Archaea and kingdom Archaeobacteria. While the typical bacteria that were previously identified, the typical prokaryotes that were previously identified, remember in this group now we are talking about organisms that have a peptidoglycan layer surrounding their embedding their cell wall. We also have typical organisms that we could identify in other organisms, but the only difference is that they were non-membrane bound. And this included all pathogenic bacteria, pathogenic prokaryotes that have been identified, and so many others non-pathogenic prokaryotes, as well as photoautotrophic prokaryotes. So the typical bacteria that you and I are familiar with are placed under this domain bacteria, and are under the kingdom Eubacteria. The third domain was also brought forth, and this domain contained organisms that had previously been described, as we have highlighted already today. And remember, in this case, we are talking organisms that were eukaryotic. That's the first thing to know about this third domain. They were all eukaryotic organisms, so they had membrane-bounded organelles. However, they could be differentiated based on their mode of nutrition and other factors. So here we have 4 kingdoms, comprising first the kingdom Protista, which included protozoa and algae. Remember, we said that these organisms are either unicellular or multicellular, and then they practice principally all modes of nutrition. So they have autotrophic, photosynthetic mode of nutrition. They have ingestive mode of nutrition, and they also practice absorptive modes of nutrition. So we have protozoa and algae under this kingdom, Protista. Then we have kingdom fungi. Kingdom fungi, I've already identified, include the yeast and the molds, the mushrooms, and most of the other fungi that you can identify. They have absorptive mode of nutrition, which is their principal mode of nutrition. Then the kingdom Plantae, which I've explained exhaustively, they are multicellular and photosynthetic, and the kingdom Animalia, multicellular and ingestive. So we now have, instead of a 5-kingdom classification, a 6-kingdom classification, but divided into 3 different domains. I will now narrow myself to microorganisms, and I will use, as you might have noticed already, bacteria as our model microorganisms. We'll narrow ourselves now to microorganisms, and we'll be using bacteria as a model microorganisms. You guys are familiar with the binomial systematic mode of nomenclature, which scientists proposed to include all organisms, or to name all organisms. So in this system, organisms are identified with scientific names that contains a generic name and a species name. So each organism is identified with 2 names. That's why the word binomial arises from. Each organism is identified with 2 names, a scientific genus name and a scientific species name. Of course, when you go back to the classification system I just put forth, you say that from domain you come to kingdom, from kingdom you come down to class, order, family, before genus. Let's leave that aside because it's not applicable in every sense in microorganisms. But I'm narrowing ourselves down on just to species and genus because that's what you are going to often use. But I'm not saying that a family or the class does not apply. In most cases, it does. So nothing has changed here. It is what you are familiar with. Organisms, two names. The first one is a genus, the second one is a species. The genus name is written with a capital letter for the initial letter, while the species name is written all in small case. If you are typing the names, you have to write them in italics as I've done. But if you are handwriting the names, you have to underline. And the underlining is done separately, not the way I've just done it. So you have to underline the names if you are handwriting. So this is the way you handwrite the names. And as indicated earlier, if you are typing, you have to do this, put it in italics. It is allowed that you could always abbreviate the genus. But remember that before you abbreviate the genus, you must have at least written it in full. Okay, you don't just start writing a sentence and the first time you're writing Staphylococcus aureus, you write as S. aureus. It's not accepted. You should have written somewhere in that same paper project as Staphylococcus aureus, so that when you write subsequently S. aureus, I'll know exactly what you're referring to. And also note that there may be instances where you have more than one organism written in the same text. And in this case, the generic name might have the same initial. I'll take, for instance, Staphylococcus aureus and Streptococcus pyogenes. If I'm to abbreviate and I just write S. aureus, which one am I referring to? Am I referring to S to mean Staphylococcus or S to mean Streptococcus? So in most cases, when you have those confusions, just write everything in full. I'm taking my time to bring out this because look, I see when students graduate from high school, they keep aside or they keep behind this principle of binomial nomenclature. You start seeing them writing bacteria in a funny shape. So take note about this. It is not allowed. Anywhere you write it wrongly, no matter the sentence that it was part of it, the whole sentence is wrong. Okay, so let's look a little bit about viral classification. Our class will end in our first segment will end shortly, so we'll need to restart. Just a reminder that immediately the lecture ends, you just launch the Zoom link. You'll be back in class within 1 minute. So viral classification is what I want to briefly talk on before we take the next section. I've tried to bring forth from 2 kingdom classification to 5 kingdom classification to 3 domain 6 kingdom classification, but it will surprise you to know that all this elaboration still does not include viruses. Viruses are still out of this story. Why? Because they are considered to be acellular. Viruses are considered to be not to show the typical characteristics of a living organism, which includes being able to undertake an independent replication and an independent metabolic system. So out of a living cell, a virus cannot synthesize its own proteins. Out of a living cell, a virus cannot replicate itself. So it needs a host to be able to undertake these principal activities that signify life. So the first anomaly in viral classification is that viruses typically do not have the 2 binomial nomenclature, binomial naming that I've just elaborated on bacteria and other organisms. So if somebody asks you what is the scientific name of the HIV virus, there are not 2 binomial names to name the HIV virus. So viruses are usually identified by common names. And these common names are either linked to the tissue the virus infects. An example is the hepatitis virus. It is named hepatitis virus simply because it infects the liver tissues. And that's the only name we have for it. The HIV virus, the human immunodeficiency virus, is named based on the clinical manifestation of the virus. In other naming, you might find people calling it the T-cell lymphotrophic virus. There's another naming for HIV because it has a tropism for T cells. Okay. At times, they are named based on the area where the virus was first isolated. You must have heard about the Ebola virus. The Ebola virus is named because of River Ebola in the Congos. It was first identified there. The Marburg virus is named because it was first identified in Marburg in Germany. That is the only name we have for it. At times, viruses are named based on the scientist who first identified the virus. And in some cases, they are named based on the clinical pathology. I've illustrated that already. And maybe so many other things are considered, but there's no typical binomial systemic nomenclature that is exhibited in viruses. The next thing I want to point out about viruses is that unlike every other organism that has both an RNA and a DNA, a virus has either. So a virus is either having an RNA or is having a DNA, but never both. So we have RNA viruses, DNA viruses, and they could be further declassified as double-stranded RNA, double-stranded DNA, single-stranded RNA, single-stranded DNA, and at times positive sense, negative sense. So those are all details that we'll be looking at in other courses. So that is it about viral classification. I'm not sure I've left out anything, but just to also add that the typical viral particle has just a nuclear material, a nucleic acid material, and a protein coat. Okay, that's a typical look of a viral particle, a nuclear material and a protein coat. But we'll identify other organisms that are much smaller than viruses, and we could call virions. Virions are nuclear material without a protein coat that can cause diseases. Prions are a protein coat without a nuclear material that can also cause a disease. So when we come back, we'll be looking at methods to classify these organisms. We have already classified them, but which methods were used by microbiologists to classify them? See you back shortly.